Hi, it's Mr. Anderson and welcome to the last AP Biology review. As you can see from the setting, I'm on the road, I'm in a hotel, but I wanted to shoot this last video as you get ready for the AP exam on Monday. I got almost 500 questions from people around the world, and so I want to try to answer the top 20 of those questions and then give shout outs to everybody. So if you're looking for just a shout out, you may want to go way to the end of this video. But the first thing I got was an overwhelming amount of thank yous from people around the world. Thanks for making the videos, and so thanks you for watching. I'm happy to hear that they've been helpful. It's always fun to see how many people are watching the AP Bio videos on the weekend before the AP exam, so I hope it helps out. Next was tons of questions about, well, how's the exam set up, and then what are some strategies that I could use. And so a lot of you are going to be really nervous, and so if we know more about the exam, that'll help. And so there are going to be two parts to the test. There's going to be a multiple choice portion, and that'll take you 90 minutes to do. There are going to be 63 multiple choice questions and then 6 gridding questions. You'll be able to use a calculator on this, and you'll probably have to use the calculator to do some of those math gridding questions. So make sure you do all 69 of those questions. Each of them are worth the same amount, and so don't spend tons of time on one that maybe is confusing. Skip ahead and just keep moving. You should take about a minute on each question. Lots of times the answer is not just going to pop out, and so my students have found if you use process of elimination, you can get rid of some of the bad answers and then just kind of make a guess. Always guess. Don't leave any blank. When it comes to the AP exam, there's no guess penalty. Last year, this is what the grid in looks like. So what you would do is write the numbers here. So if I want to put in 50, I would put a 5 and a 0 here, and then I would bubble in the 5 and the 0. Remember, a computer is going to be grading all of these, so make sure you have pencils ready to do that. It doesn't matter if I write the 5, 0 here or the 5, 0 here. It's going to be smart enough to figure that out. And they'll give you a range in which your answers could be as you're filling in those answers. And so again, I would encourage you on multiple choice to work at pace and make sure you answer all of the questions. Some of my students are pressed for time in that 90-minute period of time. Try to look at the setup, figure out what's going, and make sure you read the question. That's a big tip. Last year, you had to get a 79% or more on the multiple choice to get a 5. So AP exam, you've probably heard, is a tough test, and so you should, if you're really shooting for a 5, you should only be missing about 1 out of every 5 questions. When it comes to the free response, you'll get the questions, and then you get 10 minutes to read the questions. And so you're going to get 2 long free response, and so it should take you over 20 minutes on each of those, and then 6 of the short free response. So you get 80 minutes, so you can do the math on how long you should be taking. I would encourage you during that 10-minute period of time, just plan out the whole thing. Make sure when you're doing the free response that you're looking for those descriptive words. They're telling you, do you want us to explain or justify? If they say give 2 pieces of evidence, make sure you're giving 2 pieces of evidence. So really read the question over and over and make sure that you know what it's asking. There'll always be some mind-bending questions that are just confusing, so it's always better, again, to just make your best guess as to what the right answer is. Next one was Hardy-Weinberg. Got tons of questions about Hardy-Weinberg. I'm not sure if you mean what it is, probably how to do the calculations. I've done a video on how to do Hardy-Weinberg calculations as well. Again, what does it mean in a population that is large enough, doesn't have non-random mating, doesn't have mutations, doesn't have gene flow, and doesn't have natural selection, the allele frequencies will stay the same. So if we break this apart, P stands for the dominant allele, Q stands for the recessive allele. So let's say 10% of the alleles are for red hair. What percent would be for non-red hair? It'd be 90%. These will always sum up to 1. What do these values stand for? P squared is the homozygous dominant, so big H, for example. 2PQ would be big H, little h, and Q squared would be little h, little h. So a lot of people get screwed up because they try to solve this as some algebraic problem. And so you can think of it like a Punnett square. So let's imagine that if we go across the top, that the p-value is 0.5. So if we say it's 0.5 right here, and the Q value is 0.5, and if we say it's the same on this side, what does that mean? It means that half of the genes would be half of each of those different genes, and so what percent would be Q squared down here? You could just count the boxes and we'd find it be 25%. But we can move that value back and forth and we can figure out the P squared and the Q squared and the 2PQ. So let me give you a setup. Let's say that 9% of the individuals have red hair. What did I just tell you? I told you the phenotype. I told you this down here. q squared. And that's almost every problem is going to tell you that q squared. So if 9% have red hair, I can set q squared equal to 0.09. If I take the square root of that, now I know that q is 0.3, therefore p is going to be 0.7. Once you know that, you could figure out any of the other things. So I'd know that p squared would be 49% or 0.49, and this would be 42%. And so they're going to give you a problem since you have a calculator that's not as simple as that, but you solve it the same way. Start with the q squared, solve for the q, and then find the other ones. Chi-squared, got a lot of questions about chi-squared. What kind of a question are you going to be asked to do chi-squared? I'd bet on one of these two things. It's either going to be a genetics, like a fly genetics question, or it's going to be an animal behavior, where animals are found at different times. There was a question about that last year. What does this summation mean? It means that you have to calculate the chi-squared value for each of the different data sets in your data. O stands for things that are observed, and E stands for things that are expected. So let's say I flip a coin 100 times and I get 60 heads and 40 tails. You could do a chi-squared analysis of that. What did I just tell you? Those would be the observed values. How would you figure out the expected values? Well, if you flip a coin 100 times, you should get about 50 heads, 50 tails. And so you can use that to figure out chi-squared. You use a null hypothesis, which this is the one that I would always use. There's no significant difference between observed and expected values in coin flips or in flower types or in fruit flies. And so then you can use this chart, and this will be on that formula sheet. How do you read it? Well, if we look at heads or tails, there are two different types, heads or tails. And so if you take that -1, that'll always tell you the degrees of freedom. And so in this case, it would be 1 degree of freedom. I would encourage you to use the 0.05 value right here, and so the critical value would be 3.841. Let's say there are 4 chambers, and we're looking at how fruit flies move between those 4 chambers. Then we would simply say that there are 3 degrees of freedom. 4 chambers, 3 degrees of freedom. What would the critical value be? It's going to be 7.815. So if you get a chi-squared value that's higher than that, basically that says that something's going on, and so we fail to accept our null hypothesis. And so an example, this could be an animal behavior set. Let's say we're counting pill bugs moving back and forth. All you do is just get the mean on each side, the mean in the wet, the mean in the dry, and you can see right here that this would be my observed values because I'm seeing that in the lab, and here'd be my expected values. Next, respiration. Got tons of questions about respiration. What kind of organisms are doing those? You know, it's not just plants, but it's animals, and parts of this glycolysis go way back. So all life on our planet is using some form of glycolysis. This diagram is a little too complex. What's going on with glycolysis? We're basically breaking glucose, which is a 6-carbon molecule, down into 2 pyruvate molecules, which are each 3 carbon. What do we net out of this? We make a little bit of ATP out of this. Where's the energy going? The energy is going into a molecule called NAD, or NADH, and you can see it way back here when you get to oxidative phosphorylation. What happens to that pyruvate? It again becomes something called acetyl-CoA and then goes into the Krebs cycle. What's the big thing? Again, don't learn all these intermediates. That'd be silly. What's the big thing that's going on in here is that we're giving off that carbon as carbon dioxide. That's the carbon dioxide that you breathe out. Where's all this taking place? It's in the matrix, or in the inside of that mitochondria. Where's the big show when it comes to cellular respiration? You can see NAD right here is dropping off an electron, and it's going down the electron transport chain. Another thing that's made in the Krebs cycle is going to be FAD, and what's FAD doing? It's carrying other electrons and hydrogen. They're dropping their electrons a little bit farther down, but what are we using that energy to do? We're using it to pump protons from the matrix out into this intermembrane space of the mitochondria. All of these positive protons don't want to be there, and so as they flow back through ATP synthase, then we make ATP. Now, what are some questions they could ask you? Well, let's say we dye the carbon in glucose with some kind of a radioactive dye or marker. Where are we going to find that? Well, you should be able to figure out where's the carbon going and be able to answer application type questions like that. When it goes to photosynthesis, there are 2 parts to photosynthesis. You have the light part, which is going to be the light reaction. Where does that occur in a chloroplast? That's going to be in the thylakoid membrane. And then you have the Calvin cycle. What's going on there? That's going to take place in the stroma, and that's where we're actually making the sugars. And so just like the electron transport chain in respiration is the most important part, what would be the most important part here? It's going to be in that light reaction where we're harnessing the power of that light. How does that work? Basically, light's coming in here and here. So it's coming into a photosystem. What's a photosystem? It's a protein with a bunch of chlorophyll molecules on the inside of it. What's happening is the electrons are getting excited, and as those electrons get boosted to a higher level, they go down an electron transport chain, and we use the energy of that to pump protons to the inside of the membrane. Does that sound familiar? It's just like respiration, but inside out. What happens to eventually those protons? They flow out through ATP synthase, and then we make ATP. What are we using the energy of that ATP to do? Let me go back for a second. We're using the energy of that ATP and NADPH to add energy to carbon dioxide so we can eventually make sugars. In photosynthesis, you make something called G3P, which is a 3-carbon molecule. And then the plant can use that to do other things. We can make glucose out of it. They use cellular respiration as well, so they could get ATP from that. Or they can use it to build cellulose and to actually build that plant. One other big thing that I missed here is that electron will eventually go up and get boosted up to eventually make NADPH. What's the water used for in photosynthesis? It's donating those electrons, and those are the electrons that go through. What are we making? Oxygen, which is a byproduct of photosynthesis and luckily is a reactant of respiration. If we get to DNA and RNA, got a lot of questions about that. What's the building block? It's going to be a nucleotide, and here's a nucleotide right down here. There are 3 parts to it. You have a phosphate, we've got a sugar, it'd be deoxyribose in DNA and ribose in RNA, and then you have some kind of a base up above that. Now if we zoom into this nucleotide right here, we could count the carbon atoms. So there'd be 1, 2, 3 carbons, 4, 5 carbons. And so on this side, so we're facing up, would be the 5' end of this nucleotide. And then on the downside, it's going to be the 3'. And so how does DNA run? It just runs in opposite directions. So it's anti-parallel. And so on this side, it'd be running from the 5 to the 3. And on the other side, it'd be running from the 5 to the 3, which will become important in just a second. What's the other differences between DNA and RNA? DNA has got deoxyribose sugar versus a ribose sugar. It also has thymine versus uracil we'd find in RNA. And then, of course, a DNA is going to be a double helix and RNA is going to be a single helix. What is the cell cycle? The cell cycle is all the steps life goes through or cells go through as they divide. And so if we look at a typical cell, what's it going to do? It's going to, during the G1 phase, grow. And so the cell is going to get bigger. What happens during the S phase? It's going to copy all of the DNA that it has. And so if I have a chromosome 1 from my mom and a chromosome 1 from my dad, at the end of this S phase, you'll see the 2 chromatids of that. We will have copied all of the DNA. What happens next? It goes into the G2 phase where it continues to grow, and then it eventually goes through the M phase. What's happening then? Well, that's where it's either doing mitosis, if we're making exact copies of the cell, or it's doing meiosis, if we're making the gametes. After that, we divide the cell. That's called cytokinesis, and then we eventually have 2 cells. Now, some cells don't keep copying themselves over and over and over again, so like muscle or nerve, and so they just stay in this G0 phase. And so to understand the life of a cell, you really have to understand how does the DNA copy itself, so that would be during the S phase, and then how does it do the job of the cell? How does it make RNA and eventually make proteins and control the cell? And so let's start with the mitosis and meiosis part. So what's the role of mitosis? And these would be the phases of mitosis, which for AP Bio you don't have to learn or memorize. But what's going on in mitosis is that we are making sure that we're equally dividing all of the chromosomes inside the cell. We want to make sure the DNA goes equally into the 2 cells or the daughter cells. And so the phases, what we're seeing is if we can see the chromosomes, this would be like beginning of prophase. You can see the nucleus breaks apart. The chromosomes are now attached to the spindle on either side. So right here, this would be metaphase. You can see them all pulling apart. So we've equally pulled off the nuclei to either side. Now we have 2 nuclei, and then that cell divides in half. And so in mitosis, what are you doing? We're taking 1 cell and we're making 2 cells that are identical. When do we do this? We do it to replace any cells that are broken, and that's how you went from that zygote, that first cell, to all the trillions of cells inside your body. So then when do we ever do meiosis? Meiosis, remember, is just the production of the sex cells. So this is going to be sperm and egg. It looks a little more confusing, and the importance of that, if we look at it starts the same way. So we've copied the DNA. You can see the 2 chromatids on either side. But the most important things take place right here during prophase 1. The first one you see is that we've got crossing over occurring, and what that is you're crossing over the chromosomes you get from your mom and your dad, and you're giving your offspring a new chromosome that's a combination of those. The other thing that's happening is independent assortment. They're randomly lining up in the middle, and that's how we get those genetic differences in each of the sperm and the egg. And so in meiosis, what we have is not one division, but we have two divisions, and we go from one diploid cell to 4 haploid cells. What does that mean? They have half the genetic information of that parent cell, and they're going to be totally different from all the other cells. As we go back to the S phase and DNA replication, what happens in DNA replication? In order for mitosis to work, we have to copy all of the DNA before we can equally divide it between the two cells. And so how does that work? Well, the big major players would be helicase, and what that's doing is unzipping the DNA right here, so it's breaking it apart. We then have to add a little bit of RNA, that's called a primer. And there's an enzyme that does that. It's called primase. And then we essentially add the new nucleotides on the other side. And so if I have AAT on one side of the DNA, what's going to be on the other side? TTA. Remember, A always goes to T and C always goes to G. Now, something problematic is this. I can only add new letters on the 3' end. And so on this strand down here, DNA polymerase can move to the right. As this unzips, DNA polymerase moves to the right and adds new letters on the 3' end. That's the leading strand. It works great. But on the other side, we have to keep making these little fragments. They're called Okazaki fragments. And so we're moving away from the fork. And then when we're done, ligase will attach all of those together. And so DNA replication is important because it makes sure that we get an equal amount of DNA, the same exact DNA in each of the cells. Transcription is how we make RNA. So this would be taking place in prokaryotic cells just in the cell itself, but then in eukaryotic cells it's in the nucleus. And so what are you doing? We're copying the DNA into RNA. And to do that, it's a similar enzyme to DNA polymerase. It's called RNA polymerase. And what it does is it moves down the DNA and makes a copy of that called messenger RNA. What does that do? It carries the message of the DNA to the ribosome so we can make proteins. And so in translation, what's going on here, you can see this is the messenger RNA. It's going right through the middle. And so what it has are what are called codons. So every 3 letters of the messenger RNA codes for 3 letters of the tRNA. You can see the tRNA right here. It's transferring amino acids. And so you can see a 3-letter match right here. And then eventually it delivers its amino acid. And then we have a growing polypeptide that's coming out of the top. And so how does DNA really control a cell? It makes messenger RNA, which makes proteins. And then it's those proteins, enzymes, and the proteins themselves that make up the cell. How does an enzyme work? An enzyme is also going to be made of proteins. And so if we look at a protein itself, this enzyme, the big parts are the enzyme and then the substrate. So the substrate is the chemical that's being broken down by the enzyme or built up by the enzyme. So it's 50-50. As it docks with that enzyme, we have what's called an active site. So let me make this the enzyme. If this is the substrate, it fits in just like that. And then what you have is induced fit, where it'll actually grab a hold of it, and then it allows it to break down. And so it's the shape that's the most important in that enzyme. It allows it to attach to the substrate, put a little tug on it, lowers the activation energy, and then it's able to break it apart. How's the immune system work? This is another big question. It always starts with the macrophage itself. And so if we have something, an invader, the first thing that happens is any antigen is going to be swallowed up by a macrophage, and we now call that an antigen-presenting cell. What it does is secrete enzymes from the lysosome, breaks it up, and it's going to present the shape on its surface. Who's always helping, or waiting rather, is going to be helper T. What helper T does is sense the shape of that pathogen, that invader. Again, we're only talking about in animals at this point. And that helper T cell is going to activate B cells to make antibodies. So this part up here, the B cells and the antibodies, is part of what's called the humoral response. That's fighting the pathogens out in the body, in the humors of the body, or the liquids of the body, in the blood and the lymph and the interstitial fluids. And so the antibodies will actually adhere to the pathogen. It makes it easy for other macrophages to find it. What else does the helper T activate? It activates these killer T cells. Those killer Ts are going to find cells that are already infected and kill the infection there. And so this immune system allows us to sense a a infection and make sure the next time we're infected by that, we've created tons and tons of B memory and killer T cells, and so that we can fight that response. And so the second time you get a cold, you don't even know that you had a cold. A lot of questions about cell communication. An analogy I used in one of my videos is sometimes we need touch if we're communicating between cells. So the antigen presenting cell has to touch the helper T cell. In Gmail, what we're doing is writing from one point to another. So in the nervous system, you have one neuron and it's not quite touching another neuron, and then we'll send neurotransmitters between the two. And then a Facebook status update, if you put a status update out there, anybody could read it. So that's more like our endocrine system. So in endocrine system, what you're doing is sending out hormones, and the only ones who are interested are going to be the cells that have proteins on their surface. We call those the target cells, and they have specific receptors for that. A real common type of cell communication is signal transduction. So an example I talked about in videos is epinephrine docking with a cell in the liver. What it's going to do is it's going to release glucose from the glycogen. And so we would call epinephrine now a ligand at this surface. We call it a water-soluble ligand. What does that mean? It's docking on the surface and it wouldn't be able to move through. So if it's a lipid-soluble hormone, it would move all the way through here. But since this one's water-soluble, it docks on the surface, kicks off an alpha subunit, it activates a enzyme, which triggers a phosphorylation cascade. What does that mean? At each of these steps, we're amplifying that message. And so you can have one ligand docked on the surface, and then we can activate the hundreds, if not millions and millions of these enzymes on the inside. And so a signal transduction pathway is simply a signal up at the top docking with a receptor, and then we're sending a message into the cell. But again, that ligand could have come in and even could get into the center, into the nucleus itself, and then be turning on transcription factors and turning on genes in the center. Again, feedback loops inside us are going to be mostly negative. We have a few positive feedback loops, like birth would be an example of that, but most of them are negative feedback loops. They allow us to maintain homeostasis or keep us at a set point. So one of the most famous ones is the blood glucose feedback loop. What's going on here? The pancreas is either secreting insulin if blood glucose is too high or glucagon if it's too low. And so if the blood glucose level is too high, insulin is secreted. What does that do? It allows the liver to take in excess glucose. It really is a signal transduction pathway that opens up glut. It's taking that in and storing it as glycogen, and it's also allowing our cells to take in that glucose. Signal transduction pathway again. What happens if the blood glucose goes too low? Then we're going to secrete glucagon, and that's going to release glucose from the glycogen that was in the liver. A lot of G words in there, but allows us to keep that blood glucose level at a set point. Let's say you're type 1 diabetic. Where's your problem? We have an autoimmune disorder that kills the insulin or the beta cells in the pancreas, or the insulin producing cells. And so you can't secrete insulin, so you have to be taking insulin shots. And if it's a type 2 diabetic, then you have problems in the receptor or in the signal transduction pathway itself. A lot of questions about phylogeny, which is kind of the evolutionary history. How do you read these things? If we say here is the southern and northern minke whale, this means that they shared a common ancestor at some point back in time. And they all shared a common ancestor back here. And so each time we have a branch point, that's going to be a speciating event where we took one species and divided it in two. Now, it's not like this common ancestor here is these species. I mean, we share a common ancestor with chimpanzees, but we aren't chimpanzees. But also you'll see extinction on here. If any of these lines end, then it's going to be the end. Now, how do we build these phylogenetic trees? There's really two pieces of evidence we can use. We could use morphological evidence. That's going to be using the shape, bone structure, for example. But the big one is to use molecular evidence, so DNA and RNA. And so that's kind of a summary of the top 20 questions that I got. I can't cover everything, obviously, but I've got videos that will cover almost everything, so that might be helpful. And so I wanted to just wish you all a good luck. I promised you that I would read out some shout-outs for all of you, and so there's quite a few names you could imagine. And so I'm going to go through this. I'm sure I'll pronounce your name wrong. But in Alabama, good luck to Keenan. In Alaska, good luck to Joe and Zaquans. In Arizona, good luck to Dina and Lisa and Anthony, Catalina and Michael. In Arkansas, good luck to Lauren and Sarab. Sorry about the pronunciations. In California, you could imagine this is going to be a big list. Good luck to Madeline, Peter, Carlos, Alvin, Sudeep, Renoj, Martin, Mai, Mr. Yays, AP Biology class, or Yai's AP Biology class. Josiah, Sarah, Jason, Matthew, Abby. Good luck to Pauline, Andrew, Brian, Alvin, Darren, Bruno, Amy, Surin, Gia, Sammy, McLean, good luck. Hannah, Brian, Thea, or Thea, David, Roy, Sean, William, Ryan, Miss Arizola's class, good luck to you. Samantha, Dante, Pradeep, Michelle, Tracy, Sunny. Good luck to Mr. Holmes' AP Biology class. Good luck to Tracy, Alia, Dr. Simon's AP Bio students, Janie, California's AP Biology classes, Adam, Brandon, Natalie, Jamie, Mr. Wu's AP Bio class, hashtag GotCharFrio 2014, Judy, Brian. Good luck. Master U's Bio class, DK, Salvador, Dr. Kirchner's AP Bio class, Concho, Cameron, Tyler, Miss Shipsbull's AP Bio class. Good luck to Jonathan and Hannah and Mr. Gallagher's class, and good luck to Matthew. Good luck, California. Good luck in Colorado to Kayla and Haley and Ben and Hannah and Mrs. Keegley's AP Bio class in Connecticut. Good luck to Mr. Jorgensen's AP Bio class, Eric, Hannah, Luke. Good luck to Hamza, Mr. Karuk's AP Bio class. Good luck to Joseph and Michael and Ada and Haima and Noah and Caro and Zach. Good luck, Connecticut. Good luck in Delaware to Anudeep and Brittany. In Florida, good luck to Lev and Jacob and Brianna and Alex and Jonathan and Juan and Madison and Brian and Aditi and Tracy and Noah and Michael and Brendan and Greg. And good luck to Samantha and Victor and Aaron and Cody and Miss Klein's AP Biology class and Nadia. Good luck in Georgia to Dane and Caleb and Julie Ann and Matt and Evan and Miss Yauk's AP Bio class. Good luck to Maria, Sophia, Rosie, and Rebecca. In Illinois, good luck to Vatsal and Mrs. Sindiki's AP Bio students. Good luck to IFS AP Bio class. Good luck to Sandy Smith's Bio class, and Zaid and Adil and Daniel and Miss Sazik's AP Bio class. Good luck to Haroon and Shakib and Abdullah and Fardib and Anas and Kavita. Good luck in Indiana to Southmount High School AP Bio class. In Iowa, good luck to Ethan. In Kansas, good luck to Linda and Nick. In Kentucky, good luck to Isaac. In Maryland, good luck to Courtney and Alex and Natalie and Kenny and Toby and Jack and Kylie and Courtney and Ibican and Jenna and Stephanie and John and Salvatore and Ilian and Krista and Olivia and Brittany and Michaela and Samantha. Good luck to Aaron and Sean and Stacy and Nico and Jacob and Kelsey. Good luck in Massachusetts to Homley Soon Lee, to Mrs. Votau's AP Bio classes, to Mrs. Kashuluk's AP Bio classes, to Sid and Mrs. Meanson's AP Bio class and Mrs. Mahoney's AP Bio classes in Belmont High School, and Mrs. Lijek and Dajarini Min and Tony and Ashwin and Anatrude and Jeff and Danny and Anton and Jeff and Marwa. In Michigan, good luck to Grace and Rebecca and Ian and Sagar and Osmond and Jesse and Andrew and Ray and Jimmy. In Minnesota, good luck to Andy and Parker and Robbie and Brady and Mrs. Hammond's AP Bio class, and Haley and Ryan and Logan and Elizabeth and Mrs. Sheldrop's AP Bio class. Good luck in Missouri to Lafayette High School AP Bio class and Mrs. Ori's AP Bio class and Lulu. In Montana, good luck to all my classes, Mr. Anderson's AP Biology classes. In Nevada, good luck to Sela and Jenny and Jorge. And in New Hampshire, good luck to Brandy and Ariana and Rachel. Good luck to Mrs. Granger's AP Biology class and Mr. Grimsley's AP Biology class. In New Jersey, good luck to Eric and Annette and Anant and Aaron and Ayushi and Mrs. Lagatel's AP Bio class, and Jamie and Dennis and Rita and Caitlin and Olivia and Vraj and Ali and Micro Marath and Anthony and Timothy and Grace and Ryan and Puya and Sai and Brian and Cameron and Miss Trampler's AP Biology class, and Samantha and Mrs. Clark's AP Biology class, and Andre and Alicia and Mallory and Mina and Shay and South River High School AP Biology class, and Laura and Amir and Miss Karpinski's class, and Raritan High School AP Bio class, and Chloe and Kristen. In New York, good luck to Abby and Tenzin and David and Mrs. Jasper's Japser's AEP biology class, and Jack and Kim and Allison and John and Danny and Zoo and Caitlin and Ali and Niambi and Bobby, and good luck to Melissa and Jackie and Miriam and Reem and Dominican Academy and Lauren and Davina and Zach and Sarah and Joe and Yahudis and Mrs. Brady's class and Kim and Noah and Meklet and Mohammed and Alexander and Jorge. In North Carolina, good luck to Andrew, Kelly, Ariana and Jerry. In Ohio, good luck to Ashley and Alexi and Vincent and Maya, Zachary, Chris, Karen, Chase, Cassie, and Mr. Boyd's AP Biology class. In Oklahoma, good luck to Philip. In Oregon, good luck to Aiden. Pennsylvania, good luck to Claire and Mary and Miss Williams' AP Bio class, and Tom and Mara and Laura and Liv and Em and Reg and Lil Siegs and hashtag ribosomes and Dr. Wormer's AP Biology class, and Danielle and Nick and Upper Dublin AP Bio class. In Rhode Island, good luck to Alex. In South Dakota, good luck to Mrs. Rydell's AP Bio class, and Maddie and Gabe and Lauren and JD, Gabe and Derek and Voldemort. Man. And good luck to Eleni. Good luck in Tennessee to Sydney and Kristen and Manal and Colin and Kaylin and Ben and Mrs. Harper's AP Bio class. In Texas, good luck to Mrs. Hunt's AP Bio class, and Rebecca and Cindy and Kurtana and Kendra and Anam and Saren and Tajish and Kendall and Kamlar and Pratyoshia and Christopher and Ria and Butcher's AP Bio class. In Utah, good luck to Jake and Saul and Aaron and Sarah and Mr. Leduc's AP Bio class. In Virginia, good luck to Mary and Jay and Kashik and Davika and Paige and Afan and Cameron and Mrs. Gallagher's AP Biology class, Alana and Emily and Miss Gerald and the Inquisitive One. In Washington, good luck to Jack and Alex and Monica and Luis and Julian and Casey and Mr. Anderson and Mrs. Block's AP Bio class. In Wisconsin, good luck to Abby and DSHA AP Bio class, and Megan. In Manitoba, good luck to Natalie. In Albania, good luck to Clevy. And in Australia, good luck to Joey. In Belgium, good luck to Alishka. In Brazil, good luck to Paca's AP Bio class and Pedro. And in Canada, good luck to Atten and Anna and Hannah and Yasser. In China, good luck to Claire and Angela and Miss Kegley's AP Biology class. In Egypt, good luck to Jessica. In England, good luck to Ava and Dylan and Joe. In India, good luck to Bula and Suman and Pallavi and Anushka. In Indonesia, good luck to Sandra. In Ireland, good luck to David. In Malta, good luck to Sean. In Palestine, good luck to Anis. In Poland, good luck to Veronica. In Republic of Korea, good luck to Jay. In Russia, good luck to Alexander and Rashad. In Saudi Arabia, good luck to Majid and Samra and Rana and Rima. In Singapore, good luck to Monica and Jason and Stephanie and Rian. In South Korea, good luck to Jesse. In Sweden, good luck to Axel. In Taiwan, good luck to Annie. In Tanzania, good luck to Suedi and Trinidad, good luck to Miss O'Donovan's AP Biology class. In Uganda, good luck to Ronnie and Kitilamir. And United Kingdom, in the UK, good luck to Daniela and Greg. In the US, people not from specific states, good luck to Marty and John and Miss Lejek and Tracy and Evan and Annie and Bill and Christine and Ansley and Mira and William and Andrew and Kin and Nidalee and Liam and Karina and Alyssa and Ryan and Didon and Leandra and Rajat and Miss Barsever's AP Biology class. And then people who just said they're from planet Earth, good luck to Rashawn and the Global Concepts AP Biology class. Hey, good luck to everybody. I hope you enjoyed the shout outs. I hope the review was helpful. Good luck on Monday. I probably won't see you again, so have a great summer.